Welcome back to this course on organic chemistry in biology and drug development. In this session, we will complete the remaining coenzyme chemistry and we will show the action of a metal ion as a cofactor also. Last session, we ended up by saying a few aspects of folic acid biochemistry. We have told you that folic acid is a vitamin, is a part of the vitamin B group, and it is biosynthesized by the bacteria. So, bacteria can biosynthesize folic acid requirement, its own folic acid requirement. On the other hand, we as humans cannot do that. So, we take the folic acid from the diet, and the biosynthesis of folic acid in case of bacteria, it starts with GTP, which undergoes rearrangement and form the 7,8 dihydropteridine nucleus, and then that then undergoes pyrophosphorylation by a kinase enzyme, followed by displacement of the pyrophosphate by para-amino benzoic acid. So, the amine acting as the nucleophile, and the next step is that it is the glutamic acid which forms the amide bond with the carboxylic functionality of para-amino benzoic acid, which is now hooked up to a dihydropteridine nucleus, and then that gives rise to 7,8 dihydrofolate. Now, this 7,8 dihydrofolate ultimately goes to the this is not a double bond, this is only a single bond, goes to the tetrahydrofolate, specifically 5,6,7,8 tetrahydrofolate, and which is the active form, that is the coenzyme form of folic acid. Okay. And while in case of mammals like humans, what happens that from the diet we get folic acid. Remember, folic acid has fully unsaturated pyridine nucleus, and then this folic acid has to be reduced to the 7,8 dihydrofolate by an enzyme called folate reductase, and followed by again the similar enzyme dihydrofolate reductase to go to the 5,6,7,8 tetrahydrofolate. Now, the dihydrofolate reductase is little bit different, although the dihydrofolate reductase isolated from bacteria or from parasite or from human, they perform the same kind of reactions, but their structures are slightly different. They are, so that gives an avenue to develop molecules which specifically inhibits the dihydrofolate reductase DHFR in bacteria, or it specifically inhibits in parasite, or it can specifically inhibit the humans also. Okay. So, there are lot of interest and lot of chemistry has been developed based on the folic acid biosynthesis and this differential way of getting folic acid and utilizing it, bacteria vis-a-vis the mammals. Okay. Now, last time I also told you that the first type of drug that was developed was targeted to the enzyme which incorporates the para-amino benzoic acid to the dihydropteridine nucleus. So, based on that, this type of sulfonamides, that means para-amino benzene sulfonamide, that was that mimics the structure of the para-amino benzoic acid, and molecules which possess para-amino benzene sulfonamide moiety or can be converted into this para-amino benzene sulfonamide moiety. They can inhibit the enzyme which is responsible for incorporation of the para-amino benzoic acid. So, these are called PABA analogues, and they are antibacterial agents. Okay. And the molecules are called PABA analogues. There are different PABA analogues available, but the ultimate mechanism of action is that these PABA analogues ultimately release this para-amino benzene sulfonamide. Okay. And that when it is incorporated, the enzyme thinks that this is the substrate. In place of para-amino benzoic acid, it takes up para-amino benzene sulfonamide. So, then it cannot hook up the glutamic acid, which requires a carboxyl function at this position. So, that was the first antibacterial agent, which was based on the folic acid biosynthesis. But then people realized about this dihydrofolate reductase. I told you already that there are differences in their structure, in their affinity for the bacterial DHFR versus this is a parasite plasmodium, parasite plasmodium falciparum, and this is a mammal, maybe this is actually acting on humans. Okay. This is the DHFR isolated from human. Now, because of there are these differences, so you can specifically target one or the other DHFR. And I have shown you here 3 molecules. This is called pyrimethamine, this one. So, this is suppose A, this is A, this is B, B is the trimethoprim and C is methotrexate, that is this one, which is very having great resemblance to the folic acid structure. So, this is C. All these molecules are acting as drugs. A, that means the pyrimethamine acts on plasmodium and that is reflected in their IC50 values. IC50 is the inhibitory concentration required to inhibit 50% of the activity. Okay. So, IC50 value for plasmodium it is 0.05, possibly microgram per milliliter, that is the unit, it has got a unit, and for bacteria it is 250 and for mammal it is 70. So, you see it is extremely specific for plasmodium. So, pyrimethamine is used as an anti-malarial agent, because plasmodium is the causative organism for malaria. So, this is an anti-malarial agent. Trimethoprim is a bacterial dihydrofolate reductase inhibitor. If you look at the IC50 values, this is 0.5, plasmodium is 7. So, it is 10 about 14 times higher. Remember, lower the IC50 value, the stronger is the potency, and for mammal it is totally ineffective, 26,000 is the IC50. So, one can use this trimethoprim as an antibacterial agent, and methotrexate, if you look at the IC50 values, 0.01, this is also 0.01 and that is 0.02. So, it can act as an antibacterial agent, antiplasmodium agent, and also it can act against dihydrofolate reductase of mammals, and that gives rise to the effect. That means, it kills the cells in the human body, especially the cells which grows very fast. The cells which grow very fast, they are the cancer cells. The cells which grow very fast, they will be the major target of methotrexate. So, methotrexate is used as an anti-cancer agent. Okay. This is your antibacterial and that is the anti-malarial. So, you see that you can target DHFR specifically, it is possible, and 3 classes of drugs have been developed and they are applied, one to bacteria, another to this parasitic organisms, and the third one is the human. Okay. And all these 3 are in the is available in the market. Okay. Now, the question is that if you stop the dihydrofolate reductase from functioning, that means, if you stop the production of the tetrahydrofolate, remember what dihydrofolate reductase does, it converts the dihydrofolate into tetrahydrofolate, which is the coenzyme form from folic acid. Okay. Now, question is when you have the coenzyme, so what type of reaction it is participating in, or what type of reaction or enzyme type of, what type of enzymes it is supporting while the enzyme shows its activity, the catalytic activity. Okay. Now, I told you in the last session that actually the this tetrahydrofolate participates in the transfer of one carbon from some source to a product. So, it transfers one carbon means mostly it could be in the form of methyl, or it could be in the form of CH2OH, that is also possible. So, different oxidation level of carbon, methyl, or it could be CH2OH. So, these type of groups can be transferred. Okay. So, but these are basically all one carbon containing one carbon unit. Okay. Now, today we will be discussing only the methyl, the transfer of methyl, because that is extremely important, the transfer of methyl. Why it is important? Because you know that in the DNA that it's what is present is the uracil as the base, and in DNA it is a thymine as the base, and in RNA it is uracil as the base. So, the this conversion, interestingly, this thymine is generated from uracil, which is attached to the sugar and a phosphate, okay, and which is called the uridine monophosphate. So, that is converted into thymidine monophosphate. Okay. But this is a very important reaction. This methyl is the one which is transferred from a source to the uracil moiety, and thereby resulting in the thymine. So, this is, if you stop this, how to stop this? This it can be stopped by inhibiting the DHFR, because this is a DHFR catalyzed reaction, and the enzyme that carries out this reaction is what is called, because we are, that is called Thymidylate synthase. I will tell you what is the genesis of the name. This conversion is basically what you are having from uridine monophosphate to thymidine monophosphate. This is also can be abbreviated as DUMP, deoxy. The one I did not mention is the deoxy, because this is the sugar with the 2' oxygen not there. So, DUMP conversion to DTMP, deoxyuridine monophosphate, and remember this is deoxyuridine. So, the sugar is basically lacks the 2' OH, and this is the phosphate. So, this is DUMP converting to DTMP. This is also known as uridylic acid, because the phosphate is actually an acid. When it is attached to the 5' OH, it is O, that phosphorus, and then there are 2 ionizable groups, and so it is an acid. So, it is called uridylic acid, and this is called thymidylate acid, and since this reaction generates thymidylate acid, which is present at the biological pH as thymidylate. So, it is called thymidylate synthase. Okay. Now, this is the enzyme which is prevented from acting properly, that means the methyl transfer does not take place. Now, what happens, how this mechanism is, how this folic acid, how this tetrahydrofolate, to be specific, does the transfer, what is the mechanism of this reaction. Okay. Now, before that we have to first figure out what is the source of this 1 carbon unit. The source of the 1 carbon unit is, this is serine amino acid. So, serine, if you remove the CH2OH group in the form of formaldehyde, then you get glycine, and this is what will be called serine hydroxymethyl transferase. Hydroxymethyl transferase. So, the reaction that it catalyzes is serine converted to the glycine and formaldehyde, but this formaldehyde is very toxic. If formaldehyde is generated in the body, unless it reacts with something, it remains toxic. So, what happens that you have this tetrahydrofolate. I just write the structure. Tetrahydrofolate is in here, in this is carbonyl, this is NH2 and there is a double bond here, double bond there, and then this is NH, this is NH and this is the CH2 and then you have NH and subsequently the para-amino benzoic acid and those things. So, this is the tetrahydrofolate. The numbering was, I showed you that this is your 1, 2, 3, 4, 5, 6, 7, 8. So, this is 5, this is 6, 7, 8. That is why it is called 5, 6, 7, 8 tetrahydrofolate. But now you have to extend the numbering. Once the ring numbers are done, then this will be called the next one 9 and this is 10. So, this molecule, this tetrahydrofolate molecule reacts with formaldehyde and forms a derivative which is called 5,10 methylene THF. That means, it goes into this structure N and then you have this NH. Here is the other aromatic ring, I am not drawing that, and this becomes N and this formaldehyde is now hooked, is basically acting as a agent to link up these two nitrogens and then the para-amino benzoic acid moiety is there. Okay. So, this is called, this is 5 and this is 10. So, 5,10 methylene tetrahydrofolate, okay. 5,10 methylene tetrahydrofolate and so this is the source of your 1 carbon unit, okay. But this is not the end of the reaction. This is, so first it is the, it is a coupled reaction. Serine hydroxymethyl transferase breaks up into formaldehyde. That formaldehyde reacts with the tetrahydrofolate forming 5,10 methylene tetrahydrofolate. The next set of reactions, I think this is the mechanism of formation of the 5,10 methylene. So, the formaldehyde, this nitrogen will first react because this is more nucleophilic and then that attacks the carbonyl and that becomes CH2OH. So, this is the first step, this loses the hydrogen and the second step is this lone pair. There are two ways to define that, either the best is that lone pair goes here, the OH comes out as water and then you form this iminium ion and then the nitrogen attacks the terminal carbon, the methylene carbon and this comes to, back to the nitrogen. So, that nitrogen becomes neutral. You can draw a resonating structure like this and then also using the resonating structure you can add the this nitrogen, the 10 nitrogen 10. Okay. If you do this reaction, it actually violates Baldwin's rule because it is an endocyclization which is unlikely. So, there are many ways to do this to show the mechanism, either direct displacement by this nitrogen lone pair and kicking out. So, that will becomes an exocyclization, or utilize this type of resonating structure and bypass the Baldwin's rule problem. Okay. But this is the mechanism of formation of this methylene THF. Now, this methylene THF, what it does, this actually transfers this methylene into the this uridine molecule, and this is methylene means this carbon comes from this CH2, and in the process the tetrahydrofolate is reduced to dihydrofolate, okay. Because this is a reaction, this methylene is in the oxidation level of an aldehyde, of a carbonyl, remember that. Okay. As soon as it is converted to a methyl, that means this is reduced. Okay. So, the tetrahydrofolate tetrahydrofolate in the process is converted into dihydrofolate. So, that is a reduction. Let me again just work it out. This is a methylene, which is at the oxidation level. Here the oxidation level of the carbon is 0, and here the oxidation level of this carbon is -3. So, it is a reduction here. So, in this case the tetrahydrofolate now is devoid of 2 hydrogens. So, that is now oxidized to the dihydrofolate. So, this is reduced and that is oxidized. Okay. So, what is the mechanism? The mechanism I can draw, it is there in the slide, but better is to draw by yourself, that will make things more much more clear. So, this is your uracil molecule. Okay. And the enzyme, that is the that is the thymidylate synthase, that has an active an SH in the form of a cysteine at the active site. Okay. So, it attacks here like in a Michael type reaction 1,4 addition. So, that is the first formed species. So, you get NH and then O minus, and then you have this double bond and this N and the carbonyl, okay, and this is R, and here you have S enzyme. Now, this methylene THF, this is written in the I think what I will do, I will just write this part NH N. This is written in a just a reverse direction. So, this is N, that is your NH2, this is your carbonyl, and this is your NH. Maybe I can show it properly. So, this will be so this is the carbonyl, that is the NH. Okay. So, you do not have the other bonds. So, I have to erase this one and that one, but the nitrogen is gone. So, I have to write the nitrogen N, and this is connected here, and this is what is the methylene THF, okay, written in just the it has been inverted by 180 degree, and then you get this structure. So, N and then the benzene ring. Okay. So, now this enolate comes back and this carbon in turn becomes a good nucleophile. So, this now attacks this methylene, and the first carbon nitrogen to break is this one, because this is what is stabilized by the aromatic ring attached to the carbonyl. So, the resonance you can do up to the carbonyl. So, this is the one which is first broken, and then what you get is this. This is your NH2, this is your carbonyl, and that is NH, then N, and this is just a second, this is now released as the para-amino benzoyl, okay. Then this is glutamic acid. So, you have this attached to your that uracil moiety now. NH, this is double bond O, this is N, R, and this is the scenario NR. So, that is the this is double bond O. So, you have a hydrogen here, and your enzyme is hooked up at this position. Okay. 1, 2, 3, 4, 5, 6. So, this is the 6th position. So, at the 6th carbon, the enzyme with the help of sulfur, it is linked carbon sulfur bond. So, the first reaction is the Michael type reaction forming the enolate, then the enolate attacks through the carbon to the reactive carbon, and that makes an attack on the electrophilic methylene, and resulting in the cleavage of this carbon nitrogen bond first. And then what happens? Then your there are initially the mechanism that was suggested was this, that this hydrogen undergoes a 1,3 sigmatropic shift. Okay. So, if you do a 1,3 sigmatropic shift, then what happens? You get, this is not the actual part, I will erase that. This is the other aromatic ring. Now, this is okay. I think that is fine, no problem. This is NR, this is the NH, and this is your double bond O. So, now this hydrogen, so this is becoming methyl now, and basically you ended up with the, not yet the uracil, the thymine has not been produced yet, but the methyl has been transferred. So, now you have to expel the enzyme, that S enzyme, N R double bond O NH. So, then what happens? This hydrogen, then the enzyme is released by a beta elimination. This hydrogen is very acidic. So, some base in the enzyme active site that abstracts this hydrogen and it goes off. Okay. So, then you get what is called thymidylate acid, and in the process what you get, look at the folic acid structure. So, what is released is minus DHF, dihydrofolate. Okay. So, the dihydrofolate will again be oxidized, will be reduced to the tetrahydrofolate, and then that again participates in the reaction. Okay. So, this is the mechanism. Again I just briefly tell you, the summarize, that first it is the Michael addition of the enzyme via the sulfur of a cysteine in the active site, and the resulting enolate attacks the methylene, resulting in the breakdown of this carbon nitrogen bond, followed by the migration of 1,3 migration of this hydrogen into the to the methylene. So, making it a methyl, and this bond goes to make up the positive charge created here. Okay. Although this is in pericyclic reaction, there is no positive negative, it is a cyclic intermediate, cyclic transition state. So, this goes and that goes there. So, in one step you get dihydrofolate coming out and you get this methyl, and then there is release of this enzyme via base mediated elimination to give you the free enzyme to catalyze the next step of reactions. Okay. The dihydrofolate is again reduced by dihydrofolate reductase to tetrahydrofolate, and tetrahydrofolate participates. Okay. However, there is one problem in this mechanism, is that 1,3 shift is not allowed, thermally not allowed. Okay. And so, this is not possible thermally. So, people objected to this mechanism. So, an alternate mechanism was proposed, and I will show it quickly here. One more point is that when they put the deuterium in place of the hydrogen at that position, that means the this position, if you replace this by deuterium, then the deuterium ends up at the methyl. So, that will be CH2D. So, there is no doubt that this hydrogen is migrating, but the mechanism seems to be little bit awkward. So, an alternate mechanism was proposed, which is shown here, that this is the first adduct that is formed when this enolate is attacking the methylene. So, this carbon nitrogen bond has been broken. So, next step is basically the hydrogen. I can make it little bit more comfortable by writing the full form. This N, this one is NH, and now you have, after the first reaction, you have CH2, and then you have this uracil moiety. Okay, N R. Here it is the S enzyme. Here it is the double bond O, and you have double bond O NH. So, this is the this is the intermediate, okay, and on this side you have this para-amino benzoic acid attached. So, earlier mechanism was hydrogen 1,3 shift migrating here, but now people have suggested that the first is basically formation of the enolate. So, this is the enolate, and then this enolate comes here, this goes there, and that comes to the nitrogen. So, basically you have a methylene exocyclic methylene compound coming from the uracil moiety, and then now you do not have much problem. Then what happens, this hydrogen they are very close by, hooked in the active site of the enzyme. Now, this hydrogen as hydride adds to this. So, what you will get is again an enolate, but this becomes a methyl, and as the hydride attacks this as a nucleophile, then this becomes positive charge. So, the nitrogen now loses the hydrogen and make a double bond here. So, ultimately you will get a, that should be now that should be double bond, the plus charge will no longer be there. So, you have this enolate, the enzyme is still hooked up. So, now you bring that negative charge and kick out the enzyme. So, this is perfectly okay in terms of mechanistic organic chemistry. Earlier problem was 1,3 hydrogen shift, thermal 1,3 hydrogen shift has to be anterafacial, but anterafacial migration is not possible in a 1,3 system involving hydrogen. Remember, we are not talking about carbon 1,3 shift, that is possible, but hydrogen 1,3 shift thermal is not allowed. So, this alternate mechanism has been suggested, and most of the textbooks are having are showing this mechanism as the formation of the thymidylate acid. Okay. So, this is your thymidylate acid synthesis. So, that just to finish up, now if you have a fluorine to start with, not the hydrogen, not uracil, this is called 5-fluorouracil. So, in the body it is converted to the it is attached to the deoxy sugar and then the phosphate. So, instead of thymidylate acid, uridylic acid, you will get fluorouridylic acid, and this fluorouridylic acid will have some problem. The problem is the second mechanism, that means the correct mechanism, what you have is a fluorine here instead of the hydrogen. So, the enolization which was taking place, now there is no possibility of enolization, because there is no hydrogen, you have a fluorine, and you cannot take up fluorine as an as a positive ion, that is not possible. So, and the moreover this carbon fluorine bond is very strong. So, it is not possible to release fluorine as F plus, not even also F minus, because this bond is very strong. So, what happens, the whole thing is stuck at this stage. So, there will be the enzyme is basically now covalently linked to the dihydrofolate as the coenzyme. The dihydrofolate is linked to this fluoro version of the fluoro version of uracil, with linked up to the via the methylene to the tetrahydrofolate. So, this is a stable species, and then we can say that this is an example of irreversible inhibition of the enzyme. Okay. So, 5-fluorouracil is still used as a very good potent anti-cancer drug. So, there are two anti-cancer drugs, one I told about methotrexate, and now we see that 5-fluorouracil is also a very good anti-cancer drug. Methotrexate works by inhibiting the dihydrofolate reductase. It is a non-covalent interaction, but the binding is very strong. It has a very high binding constant, the dihydrofolate and the methotrexate. The crystal structure is all known, and it is said that basically the inhibition is essentially irreversible. Remember, irreversible inhibition is formation of a covalent bond with the inhibitor, and reversible is no covalent bond formation, it is all non-covalent interactions. But the folic acid, this methotrexate and dihydrofolate, the binding is so strong, it is a collection of many weak interactions which makes the binding very strong. So, this is called an essentially irreversible inhibitor. So, DHFR, methotrexate works by almost essentially irreversibly inhibiting the enzyme dihydrofolate reductase, and fluorouracil works on the other hand by inhibiting the thymidylate synthase, because this enzyme is the thymidylate synthase, and the mechanism of reaction says that it is perfect irreversible inhibition. Okay. So, I think we are through with the folic acid chemistry. This is very important, as you see that so many drugs have been discovered based on the folic acid biochemistry. Thank you.